All right, I think we're gonna get started. Thank you so much to all of you guys for coming. We're so excited to be with you today. Not only those of you who are here with us in person, but the over 150 people who registered to stream this webinar. We're really, really excited to talk about brain health and aging with all of you guys. I first want to thank Jennifer, Chelsey, and Molly, and all of the extension team for partnering with us for this event, and to everyone who's gathering with us from around the state. I'm Kelsey Hossabort. I'm a research development specialist at the Interdisciplinary Health Sciences Institute, or IHSI, at the University of Illinois. Our mission at IHSI is to foster collaborations and drive new initiatives that shape the direction of health-related research in Illinois. And we do this in areas that we see as critical to our community, like brain health. So part of my job, along with others at IHSI, is to partner with researchers and units from across campus and build human-powered infrastructure that supports translational research. And that clearly aligns really well with what all of you do and makes our collaboration with Extension incredibly exciting. I hope today is a great example of how we can work together and support evidence-based educational programs for the community with many other collaborative efforts and events in the coming months. Today, we're going to talk about the effects of exercise and nutrition on the brain and how we can improve things like memory and overall well-being. And this is a topic of research that's particularly important to me because it was actually the focus of my PhD research at the University of Illinois in the lab of Dr. Neal Cohen, who you'll be hearing from in just a moment. And with that, I'll hand things off to Jennifer, who's going to introduce our keynote speaker today.
All right, thank you. I'm Jennifer McCaffrey. I'm the Assistant Dean for Family and Consumer Sciences with the University of Illinois Extension. And as Kelsey mentioned, we're very excited to be offering this inaugural event. This is the first time that Extension has partnered with IHSI. So we're both on the both through University of Illinois, have similar missions. And so we thought, well, why not come together and do an event where we can highlight the current research that is going on, and then talk about what we're doing across Extension and across Illinois with our new program, the Wits Workout. So we're very excited that you decided to join us today, both in person and online, so thank you. And I have the privilege of introducing our keynote, so I'm gonna go ahead and read that. And then after our keynote, Molly Hofer and Chelsey Byers Gerstenecker will talk about our Wits Workout. So that's our agenda for the afternoon. So I'm gonna introduce our keynote. Neal J. Cohen received his PhD degree in neuroscience from the University of California, San Diego, then became a research scientist at MIT, and then a member of the faculty in the Department of Psychology and the Department of Neurology at the Johns Hopkins University before arriving in Illinois in 1990. At Illinois, Dr. Cohen is professor in the Department of Psychology, the Neuroscience Program, the Beckman Institute for Advanced Science and Technology, and the Carle Illinois College of Medicine. Dr. Cohen currently serves as the founding director of the Interdisciplinary Health Science Institute, so IHSI, supporting efforts to catalyze, facilitate, enhance, and coordinate health-related research and innovation activities across all college and research units at Illinois. Dr. Cohen's research has been instrumental in illuminating the nature and organization and brain of memory, and in advancing our understanding of memory disorders in various instances of brain injury or brain disease. His work has been central in the discovery and characterization in different memory systems, in understanding how they are implemented in the human brain, and in showing that memory can be improved through interventions involving nutrition, fitness, and cognitive training. So this ties together so well. So we are excited to hear the latest research from Dr. Cohen. So I'm gonna turn it over to Neal, who you can see at the top of the screen here, and I'm gonna turn the video off on our end.
Wonderful. Thanks so much, Jennifer. And thank you all for coming, whether in person or virtually. I understand that this is a distributed event, So thank you for bearing with me as I'm in another little corner in Illinois and not physically in front of you. Are we good for sight and sound? Okay, unless I hear otherwise,
Yes.
I'm good. So I'm going to charge ahead. So what I want to talk with you about today is the reason you should and how you might stay active to improve your memory and change your brain. A lot of the material I'm going to tell you about today is in collaboration with Chelsey. So though I'm doing the speaking, you may think of it as a collaborative effort. Okay, let's make sure this does what we want it to do. There we go. So let me first quickly give you the roadmap for the talk. I'm going to talk first about brain plasticity. Don't worry, I will define it shortly. I will talk to you about the plasticity we see in a particular portion of brain called the hippocampus, my favorite part of the brain, and I hope by the time we're done, your favorite part of the brain, and the role it plays in memory. We'll talk a little more generally about how to measure that and how to measure the health of your brain, both behaviorally and through behavior and through means of studying the brain itself. Then we get to talk about, now that we know there are methods to do that, we can come back to the hippocampus and memory and look at the effects of being active through exercise and fitness on the hippocampus and memory. We'll next talk a little bit about the positive effects, enhancing effects of making the right choices nutritionally on brain and hippocampus and memory. And then finally, one more way to be active in the sense of social networks, the effects of larger social networks on brain health. So that's the overall roadmap, and we'll come back to that after each of these very short sections. So what is brain plasticity? The brain has the ability to continually reorganize itself. It can form new neural corrections throughout life. This is what we mean by brain plasticity, the molding of the brain and the molding of behavior that occurs in response to life experiences. The really central message here is not just that the brain can change, but that you and the experiences you have can shape your memory and change your brain, which we highly encourage you to do. How do we know this? How do we know about brain plasticity? So the early scientific work comes from the study of rodents, where my colleague Bill Greeno here at the University of Illinois, one of the major figures in the work I'll tell you about now, compared the usual situation with rodents where they're reared separately in separate cages, if they're lucky with a running wheel, versus what he called environmental enrichment, putting these animals in a much richer environment with other playmates, with lots of visual stimuli, with the ability to move through the space more broadly, so action, somatosensory stimulation, a much more rewarding environment, richer environment, okay? So multisensory stimulation, permitting physical and exploratory activity and social stimulation. And he then, and others, many others, studied the effects of these kinds of environments on different parts of the brain. So exploring the effects of increased sensory and somatosensory, so body and visual input that can activate the somatosensory and visual cortices, so the orange and red portions of the brain there, increase cognitive stimulation by having animals able to move through the environment, be able to recognize objects, learn spatial maps, appreciate novelty, shift their attention, was expected to activate the hippocampus, they're in blue, and other cortical areas. And then finally, the ability for them to be able to move through space, so naturalistic exploratory movements, including fine motor skills. They were acrobats in some of these experiments that Bill and his colleagues did. That's really radically different than being reared alone with a simple running wheel, and the motor areas there in green. So the idea is you have normal environments, the standard rather impoverished environments versus these kinds of much richer environments, and then assess different parts of the brain that might be sensitive to those changes. And indeed, these parts of the brain, we're very sensitive to these changes. So on the left, a more impoverished kind of environment, on the right, the more enriched environment, and underneath you're seeing the dendrites, principal neurons in the cortex of one or another of these animals, looking at all of the arborizations, the tentacles, the dendritic arborizations of these neurons that make contact with other neurons. So the richer the arborization, the more opportunity there is to make connections with other neurons at synapses, make new synapses, make stronger connections. It is currently believed that memory is stored in these networks of connections. So the richer the network, the larger the capacity to store memory. And sure enough, enriched environment produced more elaborated dendritic arborizations, more synapses, richer synaptic networks. Now, I described that in animals, but of course, that needs to be true. We hope that that's true, and it is true in humans. Whether young in life and you get your child to move around on his own or her own and reach out and touch objects and see objects and hear objects, or as they grow older, to have more interactive kinds of play environments where there's lots of things to do and see, lots of room to move, lots of social connection. Even in video games where the social connection is virtual, kind of like right now. For me, with respect to you anyway, that can be very rich and rewarding as well too, by the time you're an adult, exploring different parts of the world and the different sites and activities you can do there. All of those have the ability to enrich the brain. So I love this cartoon from the New Yorker. A very wise and informed mom comes in and asks, which of those mobiles generates the most synapses? They do. Giving your child then a richer environment, just as giving one another a richer environment as adults, has the ability to shape our memories, shape our brains. So hippocampus and memory in the hippocampus. So I'm talking about plasticity in a general way. All of the brain remains plastic, we believe, all of your life. But there's a particular part of the brain that is a fantastic target for this plasticity, perhaps the most plastic of all, and that's the structure deep inside the temporal lobes called the hippocampus. So in the upper left, we're showing you a side view of a brain. To the left is the front of the brain, to the right is the rear of the brain. We're looking head on to the left hemisphere of the brain, but we've carved out the portion between the most lateral portions. We're looking into the more medial portions of the temporal lobe, and there we see the hippocampus. If you go down further on the left, the lower panel on the left is a real, an example of what that looks like from a brain that's individuals come to autopsy, which to the ancient Greeks sort of looked like a seahorse, which in Greek is hippocampus. Let me say that differently. To the anatomists who first explored the structure of the brain, this particular region of the brain looked like a seahorse, which in Greek is hippocampus. So henceforth, the hippocampus. Now let's take out the hippocampus and let's slice it. Let's take a cross section of it and look at what it looks like inside and look at the neurons and their connections in the hippocampus. And one of the amazing things about the hippocampus is that it continues to generate new neurons throughout life. So there's only two parts of the brain where you can continue to generate new neurons throughout life. One is in the olfactory bulb, the other is the hippocampus. That's called neurogenesis. And this is on top of synaptogenesis, creating new synapses as a function of your experience. And the image you see on the lower right is from a slice of a hippocampus, which was dyed to permit you to see older neurons and neurons that have recently divided are yellow-green, and they live alongside older neurons that are red or blue, okay? That are red or pink. Okay, so that's the hippocampus. It's a great site of plasticity, perhaps the most plastic part of all. Now let's tie it to memory. So the kind of memory we think the hippocampus is critical in is the ability to tie different elements of your experience together, to bind together into memory the different constituent elements of your experience, the who, what, where, and when of your experience. So later on, tomorrow, next week, next month, if you're able to remember some of the facts here about memory in the brain, and some of who sat alongside you, and some of who you're hearing from over the loudspeaker, who the speakers were, what kind of day it was, where you were located at the time. All of those are the constituent elements of experience that require the hippocampus in order to bind them together into a single relational memory. Let's play this out a little bit. Let's talk about what relational memory for a trip to Boston might be like. So I'll talk about a particular trip I had to Boston. I drove in over the Zakim Bridge into Boston. I was accompanied by Tom Brady and Giselle and Scarlett Johansson. Okay, that part isn't real. That isn't a real part of my memory, but imagine that those were the people I was with, first driving over the bridge and then to the public gardens, which is just a wonderful part of downtown Boston, then on to Fenway Park, another wonderful part of Boston where the Boston Red Sox play, on to Quincy Market Faneuil Hall, where we shopped and ate and listened to music, et cetera, et cetera. How do I remember the particular time and date of that? Which of those visits was it, of the many visits I had, in which I did that set of things? And the idea is that each of those elements of my experience are stored in particular parts of the brain, and then they send signals, they converge on the hippocampus to bind them together. So later, when you remember that trip to Boston, if I asked you, have you ever been to the public gardens in Boston, for example, or to Faneuil Hall Quincy Market in Boston, you might remember that little piece of information, which by then triggering the hippocampus can reactivate the entire set of features from that memory. Who you were with, how you felt at the time, when it was, etc. So those pieces, though they live in different parts of the brain, can be reactivated by activating the hippocampus. So it helps us to bind together the elements and helps us to reactivate those elements later. So that's relational memory and its dependence on the hippocampus. Now there are all kinds of relations, like remembering the order of the different pieces of that larger experience that unfolded in time, or the spatial relations among them. And memory for the spatial relations of a visit isn't like a photograph like this. It comes about by your having visited different elements along the way, having noted the spatial layout and the relations to other parts of Boston. You went from here, and then you went south to there, and then you went north to there, and then you went east to there. And your ability to tie those different pieces together to create a cognitive map of your larger environment, in this case, Boston, but in our everyday case, where we live and where we work, that ability to tie the different elements that were in different spatial locations together so that you create this spatial map depends critically on the hippocampus. And that idea about the hippocampus creating a spatial map was the topic that was the work for which several people won the Nobel Prize a few years ago, Nobel Prize in Physiology and Medicine. So temporal relations, spatial relations, all kinds of relations. All right, that's our friend the hippocampus. How do we measure its health and the health of the brain? Well, there are lots of ways to do it. We'll talk about some behavioral tests very, very quickly in just a moment. But there are ways to measure the health of the brain in humans without having to be invasive, without having to put electrodes inside animals' brains or waiting for animals to come to autopsy and look at their brains. There are ways to explore it in alive, awake, behaving human beings. And I'm giving you some images from 4 different ones here. In the upper left, functional MRI, functional magnetic resonance imaging. Many of you have know what MR imaging is, where you use magnetic field to explore the structure of the brain, and I'll get to the structure in just a sec, but you can also explore the function of the brain. You can see changes in blood flow as in response to activity of different regions of the brain. So in the upper left, what you see is several regions that are colored in are more or less active in response to some behavioral challenge. And you get to see which regions of the brain are active during memory or during attention or during movement or during thinking or during daydreaming or whatever your favorite topic is, which regions of the brain light up or become active as a result of that challenge. In the upper right, it says tractography. You can map out the structure of the brain, the different regions of the brain. And here, what you see is a map of the size and direction of the connections of different regions of the brain. So in order for the brain to do its job, neurons are connected to one another, as you see on that long panel on the right. Neurons are connected to one another and they transmit signals to one another. But in turn, collections of neurons, networks of neurons, regions of neurons are likewise connected to one another, forming tracks from one region of the brain to another. And it is now possible to characterize those tracks, to generate maps of those tracks. On the continuing around clockwise, the lower right, MR elastography. So far, we've been talking about the structure of the brain. It's possible to get measures of the microstructure, the integrity of the microstructure of the brain, looking at the elastic properties of regions of the brain. I'll tell you a little bit more about this in a couple of minutes, but it's possible to actually get measures non-invasively about the rigidity or viscoelasticity of different regions of the brain. And then finally, continuing around clockwise to the lower left, optical imaging. Instead of putting people in a magnet doing magnetic resonance imaging, it is possible to shine infrared light through the skull into the brain and record the optical activity through sensors nearby on the surface of the scalp in adults, in people our age, in kids, even in newborns. A technique that is almost unique to the University of Illinois in the Graton-Fabiani lab. Here shown with some sensors on the scalp in very happy looking small child. Okay, so those are some of the measures we can get to look at the structural integrity and the functional health of the brain. We can also, of course, look at behavioral tests. Our lab is particularly good at behavioral tests of hippocampal function, of relational memory. So the real world version of that is the one I described to you a few minutes ago. What do you remember of your trip to Boston or Chicago or Amsterdam or wherever your last trip was? What do you remember of the details of it is a way to test hippocampal function, because hippocampus is needed for you to be able to have bound that together and to remember it later. In the laboratory, we have some very, very powerful and sensitive tests. I'll tell you just about 2 of them. So we're looking in the right panel there in the laboratory. So in one kind of experiment, if you are a participant in one of these studies, you would see a series of face-scene pairings. Okay, so you see a scene, a pretty scene with a pretty face, and then another scene with another face, and then another scene with another face, a long series of those. Your job is to remember which ones you've seen before, which faces went with which scenes. So a test, so you think you're minding your own business and you're seeing lots of wonderful visual images, But what we want to know later is how well you have remembered the relations between specific faces and specific scenes. So we test that by showing you scenes later, and then a three-face array. And in each three-face array, you've seen all of the faces before, so they're equally familiar, or you've seen none of the faces before, so they're equally unfamiliar. And your job is to indicate which of those faces was studied with that particular scene. It's really a challenge, and that challenge really connects us to the hippocampus. The better you do on that test, the greater health of your hippocampus. Really the other way around, the better your hippocampus, the better you do on that test. The other test I'll tell you about briefly today is one where we're interested in your ability to recall and reconstruct the spatial configuration of a set of items. So at study time, you're shown a set of, in this case, 5 items, and these are these computer-generated creatures, really interesting objects, particularly if you're the right age. So for our younger participants, they like them a whole lot better than the faces. And you're to remember where they were located. Then there's a brief delay, only a few seconds, and then we spot you the objects. We put the objects all back up at the top. Your job is to put each object where it belongs. That is, where it was studied. And the lower two figures, what we're showing you is how we look at people's performance. So there's where the objects are supposed to be reconstructed to, and there's where on any given trial the participant reconstructed them to. And for each item, we can measure how far off they are in space. And that's a reasonable thing to do. Overall misplacement, how much misplacement is there of the objects? But it sort of assumes that each object and location are stored separately in the brain. We think of this more relationally. How well do you do at reconstructing the relations among objects, and how well do you bind specific objects to specific locations? So the measure we're most interested in is how accurate are you in putting the right creatures back in the right places? Okay, we can get an overall measure of accuracy in that, or we could look at when you swap things. So an example of a temporal swap would be if later on today you thought I spoke first, and then Jennifer, and then Chelsey, and that would be a reversal. That would be swap in time of the actual temporal locations, which was Chelsey first, then Jennifer, then me. Okay, so this is a spatial version of it. So that's the test. And I'll show you some results for that related to fitness, which improves performance on those, exercise, which improves performance on those, nutrition, the good nutrition improves performance on those. It turns out that memory and the hippocampus are important for lots of things, that this kind of memory is actually essential to navigating the world successfully, to creative thinking, as an example. So here's an example we used in an experiment. Your job as a participant is to imagine, to generate as many possible uses for a cardboard box as you can. So not just for holding stuff. What are the other possible, be creative now, what are the other possible uses of this, interesting, of a cardboard box? And if you think about how you would do it, what you're really going to do is you're going to imagine yourself surfing through, the way I think of it is surfing through your knowledge. So there are different ways you can use a box to construct things. There are different ways you can use cardboard boxes to organize where you put stuff. There are ways you can use the objects to have fun, float around in them or roll around in them. There are ways you can do it to generate objects, generate a moose or generate a desk. Okay, and you know all of these things, and what you're doing to do well on this task would be move through your relational memory of the relations among different things, the various experiences you had and the like, to come up with as many examples as you can. So how do you measure that? We just ask people to generate all the items they can. You get a fixed amount of time, a couple of minutes, and just list all of the ways in which you might use the cardboard box. And this is actually a piece of a formal test called the Torrance test of creative thinking. And in the upper panel on the left is a neurologically intact participant. In the one on the lower left, an amnesic patient. So in the same amount of time that a neurologically intact comparison participated, generated 26 different ways, creative uses of a cardboard box. One of our amnesic patients who has a normal IQ and whose impairment is seen as entirely one of memory, I mean to the outside world, to memory of a certain kind. The difference between their ability to generate creatively is enormous. And on the right is the performance above a group of patients with amnesia in the darker bar, and versus the performance of a set of neurologically intact comparison participants. As you can see, they're very, very different. Okay, I mentioned something about MR elastography. So let me just tell you a little bit more about that. This is a really complicated figure, but I think you'll find it illuminating. In the upper left panel, we have a participant lying on the bed that goes into the magnet as though we could see them from outside. So their head is inside the MR scanner. And ordinarily, we do everything we can to make people as comfortable but as still as possible so that the brain remains still while we're using the MR system to measure the brain structure and brain function. But in this case, there is a pneumatic actuator system, a little pillow that generates motion of the head. It's very, very small motion, and the motion is very regular. It's very systematic and is synchronized to when we take scans. And by moving the head in this fashion, we're moving the brain with respect to the fluid that surrounds the brain. So your brain is surrounded by membranes, and then the membranes are floating in the cerebrospinal fluid, which in turn is surrounded by your skull. So it dampens activity of your brain dampens the possibility of your brain from banging into your skull unless it's under tremendous force. We're shaking it just enough that it creates motion in the fluid, in the cerebral spinal fluid. Okay, so we're generating shear waves. We're propagating shear waves, which you see in that blue and red figure in the lower left. Okay, we're creating these shear waves. But imagine trying to, let me say it differently, we can reconstruct, it turns out, from those shear waves, what the structure of the object that's moving in it must look like, or the viscoelastic properties of the thing that's moving in the fluid, in this case, the brain. Imagine you're standing in front of a pool or a lake or a pond, and you're throwing things into the pond like boys tend to do. I'm not sure girls do that as much, but boys tend to do because they can't help themselves. So you're throwing rocks, or imagine a light rock or a denser rock or a cork. The shear waves it would produce, the ripples on the pond that would be produced by a heavy dense rock versus a much lighter rock versus a cork. Those ripples are very, very different. What we're able to do is, from the pattern of ripples, reconstruct whether it was a dense rock or a light rock or a cork. In this case, with the brain, whether the viscoelastic properties of the objects in the pond, in the pool, in the cerebral spinal fluid are relatively elastic, relatively stiff, or less stiff. And it's possible, now we move over to the right, it's possible to actually determine the viscoelastic properties of individual regions of the brain. So now we're way on the right of the right panel. We can reconstruct the viscoelastic properties of different regions of the brain, the cortex, but we're interested in the hippocampus, so we can reconstruct whether it's relatively stiff or relative or less stiff for each individual participant. Okay, create a map for each individual participant of the viscoelastic properties of the hippocampus. And then we can compare that. In this case, we'll call it damping ratio. So if you look at the figure with red triangles and a line, so the damping ratio, that measure of viscoelastic properties of the hippocampus compared to performance on that spatial reconstruction task. And we're looking here specifically as the number of times that you the participant, mistakenly swaps the location of two objects as a function of the viscoelastic properties of your brain, of that participant's brain. And there's a really striking relationship between swap errors and the viscoelastic properties of the brain. So we can use MR elasticity to predict behavioral performance here. That is, we can measure the health of the hippocampus anatomically in the MR system and behaviorally using our sensitive tests, and they're strongly related. All right, let's talk about the ways in which we can change that. Is that a fixed property of our brains? Are we born with stiff versus non-stiff hippocampus and hence good versus not so good memory performance? Or are the things we can do actively in our lives to increase the health of the hippocampus and the rest of the brain, and thereby increase our cognitive health of our memory systems? I'm sure you can guess where I'm going with this. So first, let's start with the volume of the hippocampus. So we can, when we create maps of the structure of hippocampus, when you get an MRI and you see your brain and you're shown an image from your brain, you're shown slices of your brain, but we can generate 3D maps of the whole brain and then measure, using those slices, the volume of any given region of your brain. And here we're looking at the volume of the hippocampus. And what we find is there's a strong relationship between hippocampal volume and aerobic fitness. The more aerobically fit you are, the larger the volume of your hippocampus. And this is something that's seen throughout the lifespan. So in the leftmost panel, we're looking at results from pre-adolescent children. Higher fit kids have larger hippocampi than lower fit kids. In the middle panel, we're looking at the health of the hippocampus in terms of, in terms of fitness measured by VO2 max. I'm going to show you the next slide or two. You'll see how you measure this. But the standard way, the gold standard way, is you put people on a treadmill and you measure the amount of the volume of oxygen they can move as they're doing their aerobic exercise. And what we find is the more volume, the higher the peak oxygen you can move through your lungs, the more aerobically fit you are, the larger the volume is of your left and right hippocampus. And then in healthy older adults, the same thing is true. The more oxygen you can move, the more aerobically fit you are, the larger the volume of your hippocampus. It's really quite remarkable. Here's an example of it. If you look down in the lower left-hand corner, you see someone on the treadmill looking like they're running flat out. We're measuring the oxygen utilization, measuring the peak volume of oxygen they can push out, they can move through their lungs. Okay, now as a function of the MR elastography measure I showed you earlier. So we can look at memory performance, and we can look at the elastic properties of the hippocampus. And what we find is in the left panels, the better the aerobic fitness, the higher the hippocampal elastography measure, and in the lower left, the higher the aerobic fitness, the better the relational memory performance. Okay, it's not nearly as profound a result, sorry. It's much more sensitive than the volume measure I showed you just before. So before we were looking at volume and we get these great relationships, but these relationships are even stronger if you look at the MR elastography. So just as we develop more and more sensitive behavioral tests of relational memory, we're creating more and more sensitive tests of the anatomical the anatomical measures of hippocampal integrity and brain health. A more direct way of testing these, the gold standard in tests of these things, is randomized controlled trials. So this is one done in Illinois by the Erickson group, by the Art Kramer group with Kirk Erickson, where sedentary older adults, people over 65, who were to that point in their lives couch potatoes, volunteered to be in 9-month intervention in which they were either in a walking group, that is, they met 3 days a week, every week for 9 months to exercise through walking, or they met, but they did stretching and toning, no aerobic exercise. And then they were assessed before the intervention, and they were assessed post-intervention. And the assessment I'll show you about here is the volume of the hippocampus. So there were 2 groups, exercisers in blue or stretch and toners in red. And if you look at the stretchers and toners in red, you look at baseline, then 6 months and 1 year, what you see is about a 1% volume loss in the hippocampus in sedentary people. If you're 65 and older and you're sedentary, you're losing volume in your hippocampus at a rate of about 1% of a year. Don't say I didn't warn you. But if you're exercising, the blue line, over the course of a year, you not just forestall that loss, but you gain volume also at about 1% a year. Okay, so you can actively change the health of your hippocampus structurally and behaviorally. That's just moderate exercise, and that's just 9 months, and that's in people who were sedentary prior to that. The more aerobic exercise and higher intensity aerobic exercise done over longer and longer periods of your lifetime have even greater effects on the hippocampus. Okay, let's talk a little bit about nutrition. So as interested as Chelsey is in fitness and aerobic exercise and memory and the brain, she's also very interested in, she and we are interested in nutrition, and I'm going to show you some results that Carol Boehm and then Chelsey did. Here we're looking at recall of big food inventories. Try to remember back about the kinds of foods you ate, how much of these kinds of foods and how much of those kinds of foods did you eat. And we're going to relate that to, we're going to look at particular nutrients, particular aspects of your diet, and relate it to relational memory performance. So on the left side, we're looking at omega-3 fatty acids, as in flaxseed oil or fatty fish like salmon or walnuts or soybeans. The more of that you eat in your diet, the better the relational memory performance. That's in the upper left panel. Whereas the more saturated fat you have in your diet, the more added sugar you have in your diet, the poorer the relational memory performance you have. Here I'm showing you saturated fat intake. And just to give you an example, my favorite unhealthiest meal you can get in a restaurant is from Red Robin, the A1 peppercorn burger, which has 2 patties and fried onions and bacon and bottomless French fries and a milkshake. That meal, which they sell to many people, that one meal is 3,500 calories, 69 grams of saturated fat, 6 grams of salt, sodium, 3/4 cup of added sugar. Please don't do that at home. Please don't do that at Red Robin. The more of that kind of food you have in your diet, the poorer your relational memory. Not just the poorer your heart health, but the poorer your brain health. Lutein. So I talked about a good thing to have in your diet is omega-3 fatty acids. I'm sure you've all heard that many times. Another good thing you probably have heard less about from your diet that's really important are the carotenoids, and particularly lutein, a carotenoid that you get through your diet, through green leafy vegetables or egg yolks and other things. It's an antioxidant. It has a variety of functions in human physiology. One of them is to protect the eye against ultraviolet light. So you can actually inspect people's eye to get a measure of how much lutein is in their diet. And there's a device called, or a way to test macular pigment optical density, or MPOD, which Chelsey used in her work with a variety of populations. So we can assess how much lutein is in your diet and how much lutein we think has gotten to your brain, the eye being an outpost to your nervous system. And looked at the relationship of lutein in the macula of the eye to relational memory performance. So she used that spatial reconstruction task I told you about before, where you see a set of creatures set in particular locations. Then there's a short delay. Then they're lined up at the top, and you have to put them where they belong. We're interested in particular in swap errors, in relational memory errors, and really powerful results showing the higher the MPOD score, the more lutein you've had in your diet that makes its way into your eye and the brain, the more lutein, the fewer relational memory errors you make. Okay, this is in pre-adolescent children. One last one on the nutrition front. We talked about good things, fatty acid, omega-3 fatty acid, and we talked about lutein. Now let's say one more word about bad things. An example of the really bad things are added sugar, like sugar in soda, sugar on top of donuts, sugar in your cakes and other prepared foods, versus here dietary fiber. And what she used as her measure is performance, a measure of creative thinking, the kind of measure I told you about a little earlier when we were talking about creative uses of a cardboard box. So performance on that test related to nutritional intake. And what she found is that the more added sugar in your diet, in the left panel, the more added sugar in your diet, the poorer you did on measures of creative thinking. Whereas the higher the dietary fiber in your diet, the better you did on these measures of creative thinking. So it really does matter what you eat. It really does matter how active and aerobically fit you are. Last thing, I want to give you one piece of data from other laboratories about social networks in brain health. You hear often that particularly as you age, particularly in older adults, it's really important to maintain a social network. And this was looked at in a formal way. What's the difference between living life alone in some facility versus having a lifestyle that includes lots of friends, lots of social interaction? And I want to point out that in that exercise intervention I talked about earlier, that randomized control trial that Erickson and Kramer did, those interventions involve not just walking, but walking in groups. So there's a strong social component to that. So you're active socially when you're active physically in that case. And in the enriched environments I told you about in rodents, those animals are more active by moving through space, more active by interacting with more objects, and more active socially. They had a bunch of other animals with whom they interacted. Okay, here, the social network piece of it alone is, in this case, being related to measures of cognitive function and the health of the brain in older folks on their way to getting Alzheimer's disease. So there's two panels there, and on the y-axis of each, on the vertical axis of each of those two graphs, there's a measure of global cognition. How globally intact are the individuals cognitively? And then in the upper left, on the upper right panel, there's how much pathology they have diagnostic of Alzheimer's disease. in the lower global measures of Alzheimer's disease-related pathology. In the lower panel is a particular example of pathology of the brain that's associated with Alzheimer's disease, and that's tau-related tangles in your neurons. So the accumulation of tau in your brain, a particular protein called tau in your brain, causes tangles in the filaments in the neurons, which clog them and make them less capable of working. And in both panels, if you just ignore what color the different lines are, in both panels, the more brain damage you have either globally or in terms of neurofibrillary tangles, the poorer your performance is on this global cognition measure, right? So the downward slope from upper left to lower right in each of those two panels. But in those panels, look at the solid lines. The red are ones, the red line relates to people who have high social networks. The blue one relates to people that have low social networks. And what you find is, even with brain pathology, even with increased neurofibrillary tangles, people with high social networks can hang in there cognitively, even while the brain damage associated with Alzheimer's disease proceeds. So you have reserve against the effects of brain damage if you have high social network size. Okay? So let's pull that all together. What I'm suggesting to you is that there are ways to actively facilitate brain plasticity. We have the ability to actively facilitate brain plasticity. I'm going to give you 3 slides about this, basically the same message 3 times, through things like eating well, through things like interacting with people, through things like actively gardening, being out in nature and moving around and gardening, things like moving through space by walking and other forms of exercise, by things like cognitive activity, doing new things, learning new skills, new computer skills in this case, or new language skills, and visiting new places, and sleeping well. All of those have positive effects in facilitating the plasticity of the brain and the behavioral things that result from it. Here's our friend the hippocampus, which we're treating well by making the hippocampus run on a treadmill and eating green leafy vegetables. Staying active can improve your memory and change your brain. It can make your hippocampus happier, bigger, and better able to support memory. That's the basic bottom line, punchline. The things to transmit to the folks that you interact with directly. And I want to make clear how excited I am to be able to give this kind of message to you folks who give these kinds of messages out in the community throughout Illinois. The message is the brain is plastic. There's a particular slide that I'm not seeing. The brain is plastic and remains plastic throughout a lifetime, and you can shape that plasticity. You can enhance that plasticity. You have agency in the ability of the brain to change, be plastic, support performance. That's the message. Let me close with, I gave you lots of data along the way and lots of ideas along the way, but this work, like all work, is a team sport. And there's a variety of folks on the team that have contributed to that in the memory systems lab that I run and that Chelsey is a critical member of. And really important collaborations with various groups at Illinois and elsewhere, and supported by a variety of sources like the Center for Nutritional Learning and Memory, the National Institute of Child Health and Human Development, the National Institute of Mental Health, and more. With that, I really want to thank you for your attention, and I'd love to take questions, hear comments, take questions, hear your concerns or suggestions. Thanks very much.
Dr. Cohen see the chat box too? You might want to
I'm sorry?
Okay. All right, if anybody has any questions out there, please put them in the chat box. They're checking in for CEUs right now. Have there been any studies on gluten and brain health?
On gluten? That's interesting.
Hold on just a second, Dr. Cohen.
Yeah.
Go ahead.
So while I hear the moderator, I don't hear anybody else, though I do have the chat box open so I can see written things. How would you like for me to do that?
Dr. Cohen, go ahead and answer the questions in the chat box if you can. One of the questions was, let's see, it's transferred. Has there been any studies on gluten and brain health?
Yeah, I don't know the answer to that question. I've not read anything about gluten and brain health. You would imagine looking at people of gluten sensitivity and people who don't, and the effects of that on brain health. I'm not aware. Chelsey, are you aware of any?
I'm not aware of any studies that have looked specifically at brain health.
Yeah, it's a really good question. Someone should do it.
Yeah.
Lisa, we're gonna tag you, Lisa, then. What are your thoughts on computer programs that claim to improve neuroplasticity?
Yeah, so that's a sore topic for lots of people, and let me tell you why. So we and others have done work on using video games, computer games, for a cognitive training to increase brain plasticity. The result, I think the best way to capture the results is this. Whatever the game you're playing is challenging, the portions of the brain that it's challenging can be benefited from your playing those games, but in a very narrow way. So just talking behaviorally, the amount of transfer, generalization from those games to real life is minimal, if at all. And that's the problem. So, for example, Lumosity stopped saying that playing their games makes you smarter because there, in fact, is no evidence it makes you smarter in a general way. It makes you better at the game. And they were sued and paid some fine for false advertising, and they stopped doing that. But can you get better in the areas that are being pushed by that? Yes, you can. So you might improve your working memory for those materials, your ability to attend or to split your attention, right, among the materials that you're being tested with. The hard part is making that general to all things out in the world. There's evidence, we can show evidence of changes associated with the specific learning, but not in a more general way. By contrast, the effects of fitness training are more general in lots of regions of the brain. Have I answered your question?
Yeah.
Sorry, but that's not to say you shouldn't do those things. It just says you should adjust your expectations accordingly. Okay, so doing video training of memory ability, of working memory ability, of learning names with faces, things of that sort. If that helps you in your life and your job, do it. That's great. Okay, and self-improvement is a good thing. It's real, both behavioral and brain. Understand the limits, however. In that case, you're focusing the game is focusing you and your resources in particular ways, and the improvement will occur in those very particular ways. I think in education more generally, like my job with students is to get people to think more broadly, to learn how to solve problems. That's harder to get video games around, right? The things that might generalize to all examples in your life, that's harder so far to train in video games.
Can I say something? Sure.
Please,
My
please.
father has vascular dementia. And I'm finding that, I consider him to be like on the high cognitive spectrum. And some of these things are good for people that go to a certain point. I'm about the people that are beyond that. I think what you're saying seems to be more of a proactive approach.
Absolutely. Absolutely.
So.
Did you hear her, Dr. Cohen?
Yeah, would you like me to respond?
Sure.
Yes.
Sure. So a little context first. Dementia, just so we're all clear, dementia is not is a disease, results from one or another disease. It is not an inevitable aspect of, I'm happy to tell you, it is not an inevitable aspect of aging. Unfortunately, it is age dependent, age related. So the older you are, the higher the probability of getting dementia, but it is not an inevitable thing. Of the dementias, about half of them, about 50%, are due to Alzheimer's disease, the specific disease of Alzheimer's disease. Here's another factoid. Those dementias, most of those dementias, and certainly Alzheimer's disease, start maybe 15 or 20 years before you get clinically diagnosed. So you need to understand, we all need to understand that there are the changes in the brain that eventually result in behavioral symptoms start and accumulate over time. And that's horrifying at some level because we can't detect those. You would like to be able to detect that earlier. On the other hand, I think it's motivating to all of us to ask, well, are there things I can do during that period when the effects of dementia are only beginning to occur? Are there things I can do to help overcome those changes, to ameliorate the effects, to slow the progression of the disease? And the answer is yes, all the things we've been talking about this last hour will help. So better sleeping well, eating well, moving well, engaging well, all of those active lifestyle, all those lifestyle choices actually help to, as far as we can tell, to forestall and lessen the effects of Alzheimer's disease or other dementias. I'll get to your vascular dementia in a second. So for all of these dementias, there really are things you can do. And I didn't show you much work on that, but the slide I showed you about social networks is an example. Those are people who have sustained changes to their brain. And yet, the folks who had higher social networks showed the fewest behavioral symptoms. The same thing would be true of aerobic fitness training. The same thing would be true of better eating and sleeping, okay? So there really are things we can do, but you should adjust your expectations accordingly. The sooner in time we can do those things, the better. By the time we've diagnosed them, lots of brain changes have happened, unfortunately, and that limits the window. It's much better to do it proactively. But at any time, the more of that you do, the better. For people who are much further advanced in the symptoms of vascular dementia or Alzheimer's disease, it will be harder and harder to see beneficial effects. One last thing about vascular dementia, part of the beneficial effects of aerobic exercise, as opposed to anaerobic exercise, is the increase in blood flow to the brain. And so that's it's relevant to vascular dementia as well. You would like as much blood flow to the brain as you can get. All of us would like as much blood flow to the brain as we can get, including perhaps especially people who have vascular dementia.
Thank you.
I've hold on just one second. What'd you say?
One more question.
Yeah, I think we have time. Yeah. We're going to go one live and then we'll go to some more chat box. Go ahead.
Has there been any reports on the benefits of live music, perhaps classical music? The best example would be maybe even viewing a symphonic orchestra to brain health or brain.
Yeah. Yeah. So we believe, I mean, deep in our hearts, those of us who appreciate music or appreciate the arts believe that that enhances well-being and it's related perhaps to mindfulness. It's related to getting us out of the complications and challenges of the rest of our lives, and that has beneficial effects on brain and cognition. I'm not sure there's great data on it, but I'm convinced it's true. There is some data about it. There's data, for example, that part of what's so special about music are the rhythms, and that when we pick up those rhythms, that knowledge about the rhythms, the deconstruction of the music into its constituent rhythms is a powerful thing. Is a powerful thing in people, for example, with Parkinson's disease. It's a powerful thing in people with movement disorders to pick up those rhythms and use it to guide movements. And in that regard, we know it's beneficial. I think there's another part to your question that you may have had in mind, but if you didn't, I'll put it in your mouth. And that is constantly going out and being exposed to novel things, novel pieces of music, novel orchestras, novel kinds of music, novel compositions from people who you knew but you didn't know about those compositions. That novelty, including both in the appreciating music passively and in actively trying to learn music, is very powerful. So there are people who do work on what they call Alzheimer's prevention or dementia prevention, who would argue one of the many things you should learn to do, or one of the things you should do with your time to proactively combat the oncoming effects of Alzheimer's disease, is learn a new musical instrument or learn a new piece of music. So combining novelty with your love of music would be even more powerful.
All right.
Let me answer one of the written things from the chat box. So the one that just popped up in front of me is, I mean, there are lots here, but the one that just popped up is, do I understand you to state that simple stretching moves one in a negative direction versus a positive direction for aerobic exercise? No, well, not in the way I interpret your question. Stretching doesn't move you in a negative direction. What those data say to me is that stretching is not powerful enough to stop the 1% loss you would get if you did nothing. So doing nothing, sitting on the sofa, produces the negative effects. Stretching and toning is not enough to combat those negative effects. Actively exercising, however, aerobic exercise, however, not just prevents the 1% loss you would otherwise see, but causes a positive 1%, okay? So stretching's not bad. Stretching's a perfectly good thing. It just isn't going to help you combat the effects of aging. So you need to be more active than that to combat the effects of aging on the hippocampus and on memory. I'm sure it helps. Stretching, I'm sure, helps in certain aspects of physical well-being and in your ability to move through the world and possibly in your balance. So those are good things to do. But it does not stop the otherwise relentless loss of hippocampus and memory function. Someone asked meditation and things of that sort. So we and others do have some results on mindfulness training, including meditation, that we believe to be positive and beneficial. Not as beneficial as exercise, but aerobic exercise in the things I've been talking about, hippocampus and memory. But definitely many people have done work showing that there are beneficial effects on brain function. So that's another good thing to do. I think the more active the things are you do, the more you combat the effects of dementia, the more you combat aging-related decrements in memory. But there's a variety of lifestyle choices that are overall beneficial and are to be recommended.
I'll let you choose any of the other ones you want to answer. We have about 3 more minutes.
Sure. Yeah, someone asked about, so let me take up the issue of genetic testing exactly. So the question as posed is, and I'm going to open it up, but the question as posed is there anything more people can do if they have been identified as having a genetic variant for Alzheimer's disease through 23andMe genetic testing, and how much stock do you put into those genetic tests? So that's a nice open-ended question, and I'll use my last couple of minutes on that. So what those tests tell you are about risk factors. So there's a particular variant, APOE4, which if you have, makes you 8 times more likely, many times more likely to eventually get Alzheimer's disease. And yes, you should take it seriously. Now, there's the genetic variant of Alzheimer's disease that's early onset dementia. And people get that as early as their 40s or 50s, okay? And you can see that through a 23andMe test. Then there's the age-related kind, the kind that increases in incidence the older you get. So if you're around 65, the likelihood of Alzheimer's disease is across all individuals something like 5%, but by the time you're 85, it's over 40%. Okay. Of that 5% and of that 40%, there are people with different levels of risk that come from the genetic factors. So yes, you should believe the 23andMe. It doesn't tell you you're going to get this or that. It tells you the relative risk you have of getting this or that. Yeah, I mean, in this case, it also tells you things like, premature male pattern baldness. Yep, I have that. It tells you about sensitivity to caffeine. There's actually a gene for that. It tells you about sensitivity to certain things you might eat. Cilantro. It tells you about whether you're going to the negative olfactory effects of eating asparagus. But the genetic variant about Alzheimer's disease is an increased risk factor. So what should you do if you have that? I think you should take even more seriously these suggestions about things that forestall against Alzheimer's disease. You're not certain to get it, but you're more likely to get it. That means you're more likely to have changes before diagnosis. That means it's even more incumbent, I argue, it would be more incumbent upon you to eat better and sleep better and move better and interact better.
All right, let's give Dr. Cohen a round of applause. Thank you so much for being with us today. We really appreciate it. It was fantastic.
My pleasure. Sorry I couldn't see all your smiling faces in person. Maybe next time. Thanks for taking the time, everyone.
Carla, can you hear us?
I can hear you.
All right, we're going to do this. All right, here we go.
All right.
Thank you, everybody, for sticking with us through some technical difficulties today, but you know what? We're going to roll with it. All right.
And thank you here for putting up with this for the last 10 minutes.
And Chelsey and Molly, I did get the email, so I'll be sharing my screen momentarily.
Okay,
All right,
great.
thank you.
Yep.
All right, go ahead, Ma.
Well, over the next half an hour, we're here to talk with you about something that we've been doing in University of Illinois Extension in the area of brain health and research. But before we do that, we're going to introduce ourselves. My name is Molly Hofer, and I am a family life educator with the University of Illinois Extension. My background is in gerontology. I work up here in Cook County. I cover all of Cook County as an educator. I teach mostly about adult life and aging issues within the context of families. We do cover the entire spectrum of family life. Chelsey Byers is my colleague. Chelsey?
I cover Champaign, Fort, Iroquois, and Vermilion County, so Central Illinois, but I used to cover Kankakee, Will, and Grundy, so this is not too far from where I used to be. So I do what Molly does down there.
And if you are not familiar with University of Illinois Extension, I typically say to my audiences, in a nutshell, you might think of us as a community-based arm of the Champaign-Urbana campus. We exist throughout the entire state of Illinois in communities just like this. We cover the state of Illinois, not only with family life programs, but with all kinds of different topics that help individuals in Illinois. Horticulture, we have different educators here today, horticulture educators, community health. Some of my colleagues are here today. 4-H is our youth development program. We have a STEM program, all kinds of programs. Dr. Joe Toman is our regional director. He is also here today. And we also, this is our setting, you've come to our place today, but we also travel. So if you have a need or an interest in hearing more about a topic for yourselves or for your constituents, by all means, you can contact your local extension office. We are funded through state, federal, and local dollars. Okay, so why do we teach about aging? Chelsey and I are 2 of 5 family life educators in the state. And we have taught about aging for quite a long time. I think I gave my first, or we've taught about aging forever, but I think I gave my first memory in aging talk in 2004. I've worked here for 27 years, and over again, the request, do you do anything in the area of memory? Can you talk about memory? And not necessarily related to, can you talk about memory because of dementia or the concern for dementia? Memory itself, what happens to our memory as we age? What happens to us as we age? So that is why through requests and through our own research, we started talking about it. And since 2004, we probably have reached conservatively about 17,000 people in face-to-face programming, the few educators that we have in Illinois. And Chelsey and I have been responsible for developing all of the curricula related to brain health and aging. And we've cornered the market, I guess you might say, in the area of brain health and aging. Our last series or our current series is called Brain Health, a No-Brainer. And it's a 3-part series that we do take on the road. In addition to that, I started a brain fitness exercise class, and that exercise, although we just heard how important exercise is, I started a brain fitness class on the south side of Chicago on September 7th, 2011. And I have been leading these classes with a group of older adults in that neighborhood service area with the help of 2 other educators. I do the brain health programming. We have an overriding program called Learning is Timeless, where it is an interdisciplinary model for older adults. We have horticulture programming, community health programming, book clubs, that kind of thing. But I started this brain fitness class twice a month with customized, we never did the same thing twice, customized activities, but I also had a brain health training component infused. This became so popular that our other family life educators statewide replicated the model. So we have been teaching brain fitness classes for quite a long time. So Eureka, Chelsey and I had a Eureka moment a couple of years ago. If we are doing this, why shouldn't everybody be doing this? So we designed over the last couple of years, and it is hot off the press. You have I brought a clean copy to hold up. We designed Wits Workout. Now, Wits Workout is a 24-session facilitator guide written for the lay leader. It is written with the lay leader in mind, whether it's an older adult leading a group with friends, or a church group, or a senior center, or a retirement village. And we designed this so that it could be a 60 to 90 minute face-to-face program. And we had conceptually 2 ideas in mind that are goals in mind. We wanted to use this guidebook as a form or a way, an avenue to increase socialization and potentially reduce isolation in older adults. And we also wanted to enrich intellectual engagement among older adults in settings. And as you saw by Dr. Cohen's talk, those are 2 very, very important things. Stay intellectually engaged and stay socially connected. Stay in the game of life, if you will. So those are 2 overriding goals for this program, but it is so much more. There are all kinds of things that we want to have happen with this. We did pilot test this model for the last 2 years in my south side of Chicago setting, as well as I also tested it in the southern suburbs through Bremen Township, and it was indeed tested throughout the state by our other family life educators. Now, holding that up, I'm off script, so let's see where I am. I think I'll turn it over to Chelsey. Oh, before that, this project was funded in part by Age Options, and you can see their logo, their icon here. Age Options provided a stipend to us to help with the development production of Wits Workout, and we are very grateful to them for this. They are also a site location here today, and I know they have, I think, 16 people in the room listening to this. So I wanted to formally thank Age Options before we move along.
All right, in your packet, you have a copy of the table of contents. So just because we're kind of short on time, I'm going to quickly just kind of give you a little bit overview. What we did with this book is there's 24 different units, and what we did is we highlighted certain parts of brain health. So the training components in each session have things such as short-term memory, long-term memory, recall, why do people forget, and then we really kind of did the thrust of these brain health contributors, which align with what Neal talked about in his presentation. And what our hope is that as people learn in each session a little bit about what they can do to keep themselves healthy, they'll have some behavior change. So we feed them a little bit of education, and then they do these brain activities. So they might come for the brain activities and the socialization, but then we give them a little bit of, brain health information as well. The nice thing about the way the book is written, it's kind of built like a workout. So there's a warm-up, a training component, and then there's actually a workout 1, workout 2, and then a cool down. But people can utilize it however they want. It could be anywhere from 60 to 90 minutes, depending on how many activities they do, or if people send somebody home with an activity that they can do on their own. And so, we did want to kind of just do a couple activities with you because you guys get a feel of what the components look like. So I'll turn it over to Molly here.
We also gave you one session out of the book, session 19, that is called 2 Heads Are Better Than One. And the way this book is designed, the first few pages of a session are meant as the facilitator's guide. They're meant to guide the facilitator. We actually tell the facilitator everything he or she can say or do. And any of these sessions can be modified depending on the group, the needs of the group, the desires of the group, the abilities of the group. So for instance, a facilitator can pair up individuals if some of the activities seem a little tough. The facilitator can do all of the activities as an entire group, so nobody feels intimidated or feels like they're self-conscious that they're struggling with something. When I teach Wits Fitness, I tell my groups, if you don't like this, wait 5 minutes, we'll be on to something else. And they have grown very accustomed and very comfortable with it. And I always tell people, do the best you can, and it doesn't make any difference how well you do, if you fail, because we're here to just, use the noggin. So I try to make people feel really comfortable. The training component is the research-informed part of this activity guidebook. We put in conversationally the educational information so that the facilitator can just read it aloud. He or she doesn't have to summarize or think, oh, how am I going to talk about socialization and how important it is. A person can stand up and read it. So you have it in front of you. I'm just going to read the first paragraph. The Beatles had the right message when they sang, I get by with a little help from my friends. Indeed, we all need someone we can lean on. Wait, isn't that a Rolling Stones song? If nothing else, these words tell us that social support is important. We all need social connectedness. And then it goes down and talks about why that is important and how we can remain more socially connected. So that is every one of the sessions. Because we're University of Illinois, we feel that education and sharing information that is research-based is important with the public. This is our research-based part of this to share with the public. It is largely an activities-based guide, so we definitely wanted to infuse the educational component too. Okay.
So we're going to challenge you guys with a little activity. So if you turn to your handout that says state to state, we're going to have you try to complete this activity. If you want to work with a partner or people at your table, you can.
This is the social connectedness session.
Yeah.
Highlight for the entire session is social connectedness. And in a normal group setting, if we were doing this for real, we would ask people to pair up.
Yeah.
So you can if you want. You don't
All right.
have to.
So what you have to do is identify, okay, so I'm just not going to read these, I'm just going to explain it. So of all of the 50 states, there are 4 sets, so 8 total, but 4 sets that have this, that start with the same letter, all right? But only 2 like partner pairs. So if I said I, there's more than 2 states that begin with I, right? I could go Illinois, Indiana, and I hit Iowa, I've already gone past 2, right? So it can't be I. So you're looking for states that only 2 states
Begin with
begin with a letter, all right? So
There are
only
4 sets.
so there's 4 sets of them.
You get it?
All right.
For instance, M, there are 8 states that begin with M.
Yeah.
F, there's only 1 state that begins with F. But there are 4 sets of states that begin with the same letter, where 2 of them begin with the same letter.
So if you look here, you're looking for a pair, a pair, a pair, and a pair.
Looking up how to spell a state because I cannot spell. But actually, as a child, I had to learn the alphabetical song that lists all the states. Nifty memory trick. That's how I'm doing it.
Adults too. Okay, by the way, you're not looking it up. And out in our satellite locations, and those of you individually, I hope you're also doing this.
All right, who has a pair for me? Kelsey. Kansas and Kentucky.
That is one of them.
No. Ohio.
I know, I almost did that.
Vermont and Virginia.
Yes, very good, Kath.
South Carolina,
South Dakota.
South Carolina and South Dakota.
Very good.
We have
That's the
one
third,
more
one more
pair.
set.
Tennessee and Texas. Great job.
Very
That
good.
little song ditty paid off.
Yeah.
Okay, I'm going to lead you through one more quick activity. Now, what you were just doing, you were trying to recall information you already know, right? You may not have known that Texas and Tennessee are the only states that begin with T until we just pointed that out to you. However, you have heard of Texas and Tennessee, so you were recalling information based on what you already knew. We know that recall is a noticeable change associated with aging. Have you ever said, oops, I forgot, because you didn't recall something? Or have you ever said, oh, this is on the tip of my tongue? That is, recall becomes, the difficulty with recall becomes more noticeable as we age. It doesn't mean that we can't recall, it might just take a little longer. So this is another recall activity. It is called Double Trouble. And you can pass over It Takes Two if that's the next activity in your handout. You can do that on your own because it takes too long to do here today. So Double Trouble is another recall activity. And what we're asking you to do is think of phrases that begin or end with the word double, like double trouble, on the double, body double, double talk. So write down, you'll have 1 minute. Write down as many words as you can that have the word double in it, okay? Again, it plays on the theme of 2 heads are better than 1. It can start or end. Double talk, body double, even a phrase, on the double.
Sure.
Double Bloody Mary, I'll accept it.
It's still there. I wrote
45 more seconds.
I like those. Some of my gold crowns.
Double mint gum from the crowd.
Yeah.
That's good.
Double indemnity.
Double Indemnity. Love that movie.
Double bubble. Yeah.
Double Bubble. Okay, let's come together. What are some of the doubles you have?
Double Dutch.
Double Dutch.
Oh.
Double dip.
Double dip.
Double dose.
Double dose.
Make it a double.
Make it a double.
Double bubble.
Double
Yeah.
bubble.
Double jeopardy.
Double time.
Double up.
Double up.
Double oven.
Double dip. Double,
Oven.
double oven. That's a good one.
Wish I had
That's a good one.
Yeah.
How about double digit, double cross, double duty, double standard, double door, C double, double decker, double speak,
Oh, double stuffed Oreos.
double stuffed. Very good, double tree, triple double,
boiler.
on the double. So when you go home tonight, stump your friends and family with this one. This is a good one because you can just sit at the dinner table and think of something, try to recall some of these. The last activity that we're introducing, we're taking this through, we told you this is a 60 to 90 minute session. We're just highlighting this in 10 minutes
Yeah.
for you.
We're flying through this with you guys just because we're short on time here. But all right, we're gonna do this one out loud together. So this is famous pairs. So I'm gonna read the column on the left, and I want you to figure out who we're talking about from the column on the right.
And those of you on the webinar, do it by yourselves, just think of the answers.
Yeah, or if you're online, you can feel free to type it in the chat box. Here we go. Number one, slapstick comedians in bowler hats. Hello, darkness, my old friend. This is the big one. I'm coming, Elizabeth.
Very good.
1886 Gothic novella. Dr. Jekyll and Mr. Hyde. Wherefore art thou? Corridor and whole grains. Hall and Oates.
That's funny.
Gangster couple.
Bonnie and Clyde.
Critical about movies.
Simon Ebert.
Very
Siskel and
good.
Ebert. Guess who's coming to dinner?
Tracy Hepburn.
A German fairy tale.
Hansel and Gretel.
Hansel and Gretel, very good.
Yeah.
The greatest show on earth.
Barnum and Bailey.
The dynamic duo.
Yeah, and Robin.
I've got you, babes.
Sonny and Cher.
Say goodnight, Gracie.
Burns and Allen.
Clayton Moore and Jay Silverheels.
Lone Ranger and Tonto,
Really?
very good.
44th and First Lady. Barack and Michelle. She's a little bit country.
Donnie and Marie.
Donnie and Marie. A ball of fun and her closest friend. Lucy and Ethel. Chicago siblings, Jake and Elwood.
Blues Brothers.
Cherry Garcia and Chunky Monkey.
Ben and Jerry.
Ben and Jerry.
Now, this is a very good example of taking a pen and paper activity and making it a group activity so that everyone can participate.
Yeah. So there's different ways you can lead these. You can do them in pairs. You can do them one-on-one. You can do it as a group. It's just a nice flavor. We want to just kind of highlight it. But again, the big thing we wanted to show is that we really want to promote that brain health. And so each session has a little educational piece in there, as well as to get people to increase their socialization, get people to come out, to decrease isolation, to stay engaged. You know, one thing that we have found through these programs is that at a time in life, as people get older, they might be losing friends or significant others. It helps people meet new people, right? New neighbors, new community members. And so sometimes it's just the thing that they need to get out of their homes or to come to some activity. So, you know, I think Molly knows from her programs, I know from my programs, you know, they build their own community when they come to programs like this. And so it's just something you can't even explain until you see it. So it's just something we really believe in, as well as, you know, in addition to we want to change behaviors, it's something for them to improve, you know, their lives, you know, that quality of life. That's a huge piece.
I do have older adults in my group who always ask for an extra copy of any of the activities we work on that day, because they in turn visit those that are more isolated. And that every time I teach a class, I prompt people in the audience to make sure you share this with someone who maybe wasn't able to get out today, or maybe isn't able to get out very often at all, if ever. So when Dr. Cohen put those two side-by-side pictures up of a very social group, a group that is very socially connected, and then the isolated person, we can't discount the fact that we should be the people to reach out to those who are more isolated. And I feel like I'm preaching to the choir saying this to a group here today, but you definitely, I'm sure, agree.
So real quick, Carla, I'm going to let you take care of CEUs out there in online world. But do people have questions? We do have the internet back up here, so we can look at the chat box here as well. Questions that anybody has, we kind of went pretty quick because of our little.
Awesome. I love the way you have it laid out and everything.
I also want to tell you the activity pages are on one side and the answer is always on the other, so that the facilitator doesn't have to go to the back of the book. Or
That's great.
and I think I said it already, but it is designed so that any activity pages are designed to be duplicated. The book itself is copywritten, but the activity pages are designed to be duplicated. Yes.
Order of units meant to be done sequentially?
Good question. No. The theme, each session has a theme, and we tried to base the theme. Some of the themes might be done at a certain time. One theme is called Happy Birthday USA, for instance, so you might want to do that in the summertime. But you can take them out of sequence. If you as the facilitator think, oh, I'm going to talk about socialization today, so I'm going to take this activity from here and this one from another socialization activity. I don't know if we said this, but there are 12 educational components, and they're repeated twice. So that this is meant to be a 2-year, it has a 2-year shelf life if done monthly. And someone said to me, well, then what? And then I said to them, you start it over again, because you'll have a new group all the time of people coming in. Other questions?
We have a couple here on Zoom. Somebody says, where are these offered? I would like to share with my mother. And then do you run into any issues with educational level and or ethnicity with this program? I'm going to let you talk while I type something in here.
Yes, we have run into issues. That's why we pilot tested it, and it's not foolproof. We in turn may, as we use this, with other groups, we may have people come back and say, you know, that doesn't resonate with the group that I work with. And that's why our materials are always updated. And the group that I worked with for since 2011 is an all African-American older adult group. And the group that I pilot tested this for the last 2 years is in the southern suburbs, a very diverse group of different backgrounds. So that was part of our pilot testing, but you know, we may come across other situations. It is not in Spanish. If you're thinking that is another issue. It is not in Spanish. And if we do translate it into Spanish, we have many experts at University of Illinois Extension who can help us make it culturally significant.
And it was also pilot tested in like rural communities in Western Illinois, also in Southern Illinois. So we did try to do different parts of the state when we did the pilot before we put this in print. And we did have to change a couple of the activities. So that is something that, you know, some of the things just didn't fly. So that's how you learn.
Or I could use a phraseology, and someone in the group might say to me, I have no idea what that means. And in turn, when some of my groups were giving answers, they would say something and I would say, I don't know what that means. So as you know, you come across that in your day-to-day interactions with your groups anyway. We learn from each other.
Do we just order them online or do you actually have them available?
We have them available here in person today. For all the people out there, you can order them online. We also have them in print or on USB drives, depending on how people like things.
And as far as pricing, the hard copy book is $75. The thumb drive is $75. If you want both, we have a combination price of both for $115. So, and today, we have them here so that there isn't any kind of shipping cost. Any other questions?
Question about copying or the handouts, maybe you want to mention how people can.
Yes.
So the handouts can be copied, but we ask that people don't copy the entire curriculum. It is copyrighted. Is that a word? Copywritten? Like, that is so correct. I'm under stress because of all the tech issues. Please forgive me. So yes, it is copywritten, but we have given permissions for all the activities to be copied. That is the intent. We want you to copy these for your participants. So once it's purchased, you can copy it for your participants over and over again for each of your groups that you lead.
And all of the activities are organic. We created every activity. So they weren't adapted from someplace else or with permission. We created every activity organically. Yes, sir.
I'm hoping to do this as part of my public library senior programming. Does it come with any kind of like graphic images that you can use in promotional flyers or anything like the logo for this?
That's a good question.
That's a good question. If people didn't hear, somebody asked about the logo for promotion. Feel free to email me. I was the one on your registration, clbyers@illinois.edu, and we can help you with that. So,
are you part of the Age Options Library? Yes, I will be providing training also through Age Options. Yes, yes.
I know this area and we are teaching. Is there any way we could sit in on one of the classes or could somebody sit in on one?
Oh, absolutely. Absolutely. Talk with me after and I can share with you. I have ongoing programs still at Bremen Township and very ongoing with our Learning is Timeless program in the city. We've been doing that for many years. Okay, terrific. Absolutely. You can participate.
we hope it's very affordable. We would like them to get out and be used. So.
Awesome. And it's beautiful the way it's set up. It's awesome.
All right, we want to be mindful of everybody's time. If you did sign up for CEUs, please sign out on your way out to make sure you get your CEUs. Thank you everybody for coming and being patient with us today. Thank you.
Of course. Yes. That's a lot of work. And thumb drive. Oh, right.
No, but I got it. I'll just get reimbursed. Oh, I got a credit card. No, I don't.
Get an extension square reader.
I know. I need to go to Hofer for checkout.
Okay, what would you like me to do?
Or actually, yeah, if you could, if I give you this.
No problem.
Yes.
Thanks for, I'm glad you're able to come and meet technology work for them.
I'm just gonna pull up the registration if people want to buy it and pay with credit card. You can just run it through, and then I can help answer questions.
Okay. Yeah, I can. What's your program coordinator?
Emily.
Emily, I'll go sit over by Emily.
Okay, and you can just fill it in. It's just ready to go. It's a form.
Okay.
Yeah, I mean, I'm gonna, what my plan is to share, I'm gonna share a PDF of the slides with Chelsey and then she can discuss it.
Yeah, and
She'll email it.
recording too.
It'll be a Neal recording, not ours, that's okay.
Yeah, we'll clean it up too.
So she was on recording.
Oh, yeah, that's
And they're not too sure when it.
yeah.
So
Oh, yes, definitely. Happy to share. You'll have his talk, you'll also have a PDF of all the
No, they don't, most home health does not.
So we just love to be able to figure out how to do more like direct connections.
So those of us that are still online and asking questions about CEUs, I'm just going to share that you will receive an email from Continuing Education Institute of Illinois with an evaluation, and to get your CEUs, you will need to complete that evaluation and return it to them, and then they will email your CEUs. So if you have any other questions, feel free to catch me on the chat box.
That's my, our director for research and information on the front, because I tend to get